Lantus, insulin glargine for millions of diabetics worldwide. Its production requires the seamless interaction of many individual processes. Over the next 10 minutes, we'd like to take you on a journey to discover all about insulin. Our journey begins in 1921, when two Canadian researchers isolate insulin from the pancreas of cattle. This classic natural material synthesis marked the beginning of insulin therapy. In 1983, researchers at Herx developed a biochemical method allowing the production of human insulin from pig insulin. The 1990s marked the beginning of the era for the biotechnological production of insulin. The state-of-the-art plant for producing Lantus was commissioned in 2000. In Frankfurt today, there are various facilities for the production of a range of different insulins, the sterile pharmaceutical product along with insulin pens. In short, everything right up to the finished product for the patient. Insulin is a protein, or more precisely, it is a hormone. It's made up of amino acids that are threaded together in a specific sequence and which are also in part linked with each other. A glimpse at the 3D structure goes to show just how complex insulin really is. Lantus is a human insulin analog, which is completely soluble in slightly acidic solution. When Lantus is injected into subcutaneous tissue, it's neutralized, leading to formation of microprecipitates, from which small amounts of insulin glargine are continuously released, providing a smooth concentration time profile with a prolonged duration of action. This is the key factor in the operating principle of Lantus. In producing insulin, we have billions of little helpers. Escherichia coli bacteria, or E. coli for short. They contribute to our being able to have insulin such as Lantus today. This is because we've genetically modified the bacteria so that they produce a precursor of insulin. Then it's our job to extract this precursor, to fold it, cleave it, and purify it. It's a process that requires a lot of time and experience. We start with the fermentation, which is the propagation or cultivation of the E. coli. Crucial in this, and it remains our trade secret, are the nutrients and the right temperature. In other words, everything that improves the well-being of the bacteria in the culture medium, and that encourages them to continue to reproduce over and over again. After inoculation of the lab flasks, there's the 2,000-litre pre-fermenter, and then the gigantic main fermenter with its 60,000 litres. At the end of 2 days at 37 Celsius, it's transferred to the harvesting vessel. Then the processing can begin. The bacteria produce a precursor of insulin. This fusion protein is like a pearl necklace on which the individual pearls have been correctly threaded. But to get at this protein, we need to destroy the cell wall of the bacteria. For this, we subject them to a short but extremely high pressure. The bacteria cell walls are unable to withstand this sudden shock, and they burst. Now the fusion protein is isolated and purified before beginning the folding process. What this means is that we need to provide the conditions to allow the protein to take on the correct 3D structure. But there's more to it than that. The superfluous parts of that pearl necklace also need to be removed or cleft. Most of all, it's these folding and cleaving procedures that are the critical points. They determine the yield and the purity of the end product. At this point, we have, for the first time, insulin with a purity level of roughly 60%. The plant for the production of insulin glargine is controlled by staff at the measuring station. They regularly take samples for lab analysis and ensure that the process is running optimally. A job that requires a great deal of experience. The ultra-purification of insulin is achieved through multiple steps using chromatographic methods, in which there resides a great deal of company know-how. The chromatographic purification of proteins is, in itself, an established procedure, but as so often, it depends on every nut, bolt, and screw. At the end of the initial chromatographic cleansing, we have an active ingredient with a purity of 90%. After the second purification, that rises to over 99%. By crystallization, the active ingredient is then isolated, recrystallized and filtered, all under cleanroom conditions. Subsequently, there's freeze drying, the filling into stainless steel containers, and cold storage. These many steps in production result in an extremely pure, white, fine crystalline powder. Crystalline insulin is of little use to a patient. Swallowed in tablet form, it would simply be digested. That is why insulin needs to be injected. For this to happen, the active ingredient needs to be dissolved in a liquid and put into ampules or bottles. This needs to be carried out under conditions of the highest sterility. Carefully and bit by bit, the crystalline active ingredient dissolves and results in the liquid form of insulin we know as Lantus. However, before the ampules can be filled, they need to be thoroughly rinsed, sterilized at 270 Celsius, and transported to a hermetically sealed high-sterility room. It's there that the filling of the ampules takes place. If any manual intervention should be necessary in that area, it can be done only by using the fixed installed arm-length gloves. Not until the insulin ampules are completely sealed can they leave that area and move on for intensive checking, followed by packing. The pharmaceutical drug, the Lantus ampules, can now be installed in the insulin pens. Since 2007, Frankfurt has had a production facility for the manufacture of our in-house developed insulin pens. Our principal product is the disposable pen, the Solo Star. Naturally, we also have a range of insulin pens. The Lantus Solo Star is a high-tech product. It comprises a mechanical plastic section, the insulin ampule, and the outer casing. The components are assembled in a process of 11 steps. On the production line, over 6,000 checks a minute are made, involving 14 cameras and 500 sensors. The final checks are followed by storage in high bay racking. Along with all our other high rack storage areas, it has a large cold area for storing the temperature-sensitive insulin. Now the real logistics begins. In chilled containers, our insulins and insulin pens make their way to over 100 countries, packed in more than 1,500 variants. The journey from bacteria to the finished insulin pen is a long one, a journey that requires a lot of expertise. All made possible first through the knowledge and experience of our workforce. The manufacturing process of our insulins is unique. Because it's the process that determines the product, others can only produce a drug which is similar to the original, but not identical. A brief look at our integrated production location is enough to show why, at the beginning of 2010, Frankfurt was chosen as the location for the group's worldwide diabetes division. Here there is research, development in the biocenter, active ingredient production, the production of sterile pharmaceuticals, right up to the production of the medical device, the pens and distribution. everything surrounding the topic of diabetes. Here we research, develop, produce and market the products, helping to bring health and an improved quality of life to those millions of people affected by diabetes. We shoulder this global responsibility each and every day.